Telomeres and telomerase. This is a fascinating subject, you guys. So there's some good diagrams in here. Let's just talk about it a little bit before. So if you just think, like, the easiest way I think to think about chromosomes is just think about a chromosome in an X shape. Obviously we have X and Y chromosomes, but you think about the X chromosome. So telomeres, if we're looking at the X chromosome, if you just think about like little caps on the tips of each X or on the tips of each line, those are the telomeres. So in general, the longer our telomeres are, the more times our cell can replicate and the more health span the cell has while it's going through its Hayflick limit of replication. So the Hayflick limit is just, it seems to be a predetermined thing for certain cells that tells them they can only replicate X amount of times and then they're going to die. And then there's pluripotent stem cells, which have different types of things that they can do. Telomeres are repetitive nucleotide sequences that form like a little hat or cap on the end of the chromosome, and they maintain the chromosome's integrity. They protect it from damage, and telomere dysfunction leads to significant health problems. One very, very interesting thing to keep in mind, you guys, and Brett Weinstein is a great evolutionary biologist to listen to. I really like Brett. I would love to come on his podcast and talk to him about some of this stuff. In one of Brett's I think it was in his PhD thesis, he discovered that because lab mice are so inherently inbred, that they have extra super duper long telomeres, which makes lab mice, so just think about this, you guys. The lab mice that we're testing, that the pharmaceutical companies are using, and they're using these lab mice on purpose. They know this stuff exists. They're not dumb. They're developing drugs. The pharmaceutical companies on purpose are using lab mice that have super extra duper long telomeres, which makes those lab mice super resistant to disease. So if we're testing drugs on mice first and then on humans, if we're testing the drugs on mice with super duper long telomeres, they're going to be way more resistant to disease than humans are. And it's not surprising, especially looking at the last 5 years, if you test stuff on a couple of animals and then you decide that is good enough to test it on 80% of the world's population, really bad stuff happens, and that's what we're seeing right now. So telomeres are very important for our ability to maintain our health. And a lot of different things can shorten telomeres, like just chronic mental stress will lead to dysfunctional telomeres and shortening of telomeres, dysfunctional amounts of nutrients and supplements in your body. If you don't have enough stuff to produce telomerase, the proteins and the genes that make telomerase, our telomeres get dysfunctional. Mice with dysfunctional alleles or different variations of the telomerase gene, they've helped show researchers how important the telomeres is. So if you mess with the telomerase genes in mice and you make them dysfunctional, they die, and they're way more sick. They're way more prone to chronic inflammation, neurodegeneration, diabetes, autoimmunity, all that stuff. And again, just chronic mental stress can shorten your telomeres. It's kind of like with all this other stuff. If you can do a bunch of stuff in your life as far as changing your lifestyle and putting the proper things into your body and not putting bad things into your body, you can change the epigenetic mechanisms that are happening in our body, and you can literally change the way that your DNA is getting expressed and to what extent, and you can significantly change your own health. So we have way more power over our own health than mainstream medicine wants to give credit for, and there's numerous reasons for that. But if you understand if you understand this stuff exists in the first place, which most medical practitioners don't, because this is not taught in medical schools for some reason. None of this stuff is taught in medical schools. It may be briefly touched on, that's about it. If you know this stuff exists and you understand how it works, you can do things for yourself and for your patients to significantly help them just live a better life and be more healthy, which is what I'm trying to do. As telomeres shorten, cells basically lose. I just think of the telomeres as the chromosomes army. You have like army defensive units on the ends of your chromosomes, and as you lose people or as you lose parts of your army unit, or as they get killed off, or however you want to think about it, as the telomeres shorten, chromosomes lose their ability to fight off disease, they lose their ability to stay healthy, and the cell in general loses its ability to function. So again, there's specific peptide therapies that significantly increase the amount of telomerase and that actually lengthen telomeres. NMN is not a peptide therapy, but NMN and NR are two that have been shown to increase telomerase and telomere length. Increased NAD+ helps improve telomere function and length. And sirtuins help improve telomere function and length. And a couple of the peptides that we mentioned before, MOTC especially, and another one called epithalin, those are mitochondrial peptides that help improve telomerase activity and telomere length and the health and function of the cell. B vitamin derivatives are very, very important for telomerase production. Again, NMN, all this stuff. So all of this stuff in all of these classes, you guys, the vitamins, the nutrition, the peptide therapy, the information about the epigenome, you need to understand all these different parts of it for it to make sense with each other. Like, you can have some understanding if you understand like what telomerase is and what mitochondria are, but you're not going to have a comprehensive, in-depth grasp of it unless you understand all of these things in relation to each other. And that's what I've been trying to do in these classes. Just remember, shorten telomeres, more unhealthy cells. As the telomeres shorten, your cell's lifespan is going down, your lifespan is going down. So as far as senescent cells, you guys, one of the things that happens as we age, like I've mentioned before, is we lose energy. Our cells lose the cellular mitochondria lose the ability to properly produce ATP, and we start to go into significant states of energy deficiency, which leads to a huge amount of age-related diseases and just aging in general. Telomere shortening is one of the most significant controllers of age, health span, and lifespan that we've found in the human body so far. It seems to be the case that if you increase cellular or chromosomal telomere length, the cells can replicate more and longer, and theoretically, if you can keep improving telomere length so the telomeres never shorten all the way, you can make immortal cells. And there's some jellyfish that seem to be able to do this, you guys. So this stuff does happen in nature. It is possible to do it to human beings. And this is one of the things that people are trying to figure out. So I'm all for trying to like regulate our telomere length with nutrients and supplements and food and stuff like that. What I'm not for right now, at least, and not anytime in the near future, is messing with the actual DNA and RNA, because we have no idea how that stuff is working and it's just not safe. But we can mess with it on different levels by putting the proper nutrients and vitamins into the body. We can improve the length of our telomeres, which makes our cells healthier. It makes them live longer, which makes us live longer, which is the goal, and healthier. Another thing that senescent cells do, you guys. So senescence is just aging. As cells get older, as the telomeres shorten, they age, they go into senescence. When senescent cells start to accumulate, bad stuff happens. This is insane, you guys. This is why biology is so cool. If you look at animals like fish, for example, let's just take salmon, because I was a fly fishing guide for a decade. One female salmon had thousands of eggs in them. It's amazing. It's this massive skein of eggs. What they've been able to show with animals that have eggs and that have more than one egg is if one individual egg gets infected with a bacteria, let's say, before the actual bacteria itself gets into the other eggs already start producing defenses against that bacteria, just like the cell that's in the egg that's infected with the bacteria does. So if you have 10,000 eggs and one egg gets infected with bacteria, before the bacteria can spread to the other eggs have already sent somehow from that one egg. They've already been told by that one egg that something is wrong and that you need to make these proteins and these enzymes to help you stay healthy and to fight off this thing. So that's insane. And that is exactly how the cells in our body are working, you guys. We have trillions of cells in our body. They're all communicating with each other. Just because we have an area, like say we have if you injected something in your hand, your brain is going to know about it almost immediately. And that has to do with like nerve conduction and that stuff, but it also has to do with cell to cell communication. And we know this stuff happens for a fact. We don't understand this really at all, but this is one of those things. It's like, how do the cells know to tell each other? Or do they actually know? Or is it just like an electric potential thing combined with chemical potential and osmosis? Is it a different dimension? I mean, these are things that people are thinking about. It's very, very interesting to me. Overall, if your telomeres are shortening, you're going to have more old senescent cells close to each other, and they're not going to be able to communicate properly. So it's not just a problem with the cells themselves getting old and decrepit. It's when they get old and decrepit, they can't communicate with each other, so they start not only affecting that specific area, but everything else, and that leads to body-wide dysfunction, which is one of the things that autoimmune disease is, you guys. Chronic sympathetic hyperactivity, again, is one of the hallmarks of aging. It's basically chronic inflammation. It causes all the hallmarks of aging, and it's one of the hallmarks of aging. The vagus nerve is the most parasympathetic dominant structure in the human body. It's the 10th cranial nerve. They call it, in Greek, I think, the wandering nerve. It is a nerve that comes out of our brainstem. It connects all of our organs to themselves and to the brain. And it's the primary connector between the gut-brain axis, or the enteric nervous system, and the brain, which is super duper important to produce things like serotonin, dopamine, all sorts of other things. Remember, you guys, we have more bacteria in our gut than we have cells in our body, and the bacteria produce a huge percentage of our neurotransmitters like dopamine and serotonin, and they affect a huge amount of other things that really, really affect mental health and physical health and vice versa. So dry needling, electrical vagus nerve needling, is one of the best ways to stimulate the parasympathetic autonomic nervous system, to reduce sympathetic hyperactivity, elevate parasympathetic activity, and achieve autonomic nervous system homeostasis. Exercise does the same thing. Supplementing and properly eating does the same thing. Peptide therapy does the same thing if you do this stuff correctly, which is what this class is allowing you to do. So in these classes, you guys, that's what I'm basically trying to do, is give you the means and the information to do this stuff for yourself. Here's another good diagram of the telomeres. The telomeres, you guys, are the colored ends on the chromosomes there. The bigger part of color on the end of the chromosome, the bigger the telomere, the longer the telomere, typically more healthy it is. There's telomerase, which is an enzyme that helps with telomeres. There's TERC and TERT. There's two different enzymes and ways that the telomerase are getting regulated, and there's lots of different ways that we don't understand. But basically, telomere shortening leads to inflammation, oxidative stress, toxic cell death, and inhibited ability for the cell to renew itself, or to keep itself healthy, or to replicate in the future. Once this telomeres start to shorten, the Hayflick limit is reached. So the Hayflick limit seems to be a predetermined number of times certain cells can replicate. So there's some cells that can infinitely replicate, like stem cells, and there's some cells that cannot. As the telomeres shorten, you're getting closer to the Hayflick limit, or each time the telomere shortens, you're losing one time of being able to replicate the cell. This activates DNA damage, it activates cell death, but it may not be properly programmed cell death. And then if you can increase telomere length, you guys, that leads, so telomere shorten, decreasing cells lifespan, health span, and ability to replicate or reproduce, more or less. If you can increase telomere length, it does the opposite. It allows continuous proliferation and cell survival. It allows escape from replicative senescence. Increasing telomeres is basically like giving immortality juice to genes, or to cells, or to chromosomes. Increasing telomere length helps keep things immortal if you can do it in a proper way, and there actually are ways to do this now, you guys. I believe he's from Japan. He won the Nobel Prize. They're called the Yamanaka factors. There's these 4 molecules that if you put them in the proper concentrations with each other, you can basically reverse, like my current 41-year-old heart cell. If you put those 4 factors in it, you could reverse those heart cells all the way back to pluripotent stem cells that could turn into anything else, which is basically taking them from an aged cell back to like an extremely young new cell. The problem is we don't know how to control that that conversion. I always think, kind of funnily I guess, is that like, if you put that stuff just into a human being, would they just melt into jelly because all their cells would turn back into pluripotent stem cells, or what would happen? But these are the things that are being looked into in science right now, so it's good to be aware of all this stuff. Here's a good one on other things that affect telomere length, you guys. So it's not just aging, like telomeres get shorter as we age in general, but we can do things to make them longer as we age. So antioxidants, vitamin C, vitamin E, polyphenols, those are all talked about in our nutrition and supplementation class. Anti-inflammatory agents, omega-3 fatty acids we talked about, decrease in chronic inflammation, oxidative stress, and inhibit pro-inflammatory pathways, which is what eating well does more or less. Peptide therapy inhibits pro-inflammatory pathways, reduces oxidative stress, reduces telomere shortening, improves telomere length. There's a bunch of different ways that we can do this. Supplementation and peptide therapy. So I've talked about this a couple of times, you guys. This is a good slide to illustrate the difference between mitochondrial and nuclear substances and genetic material. So there's mitochondrial genetic material and nuclear genetic material, and they communicate with each other. So reactive oxygen species and other things like that, like proteins and cytokines, can lead to either improved regulation and communication between mitochondria and the nucleus, or decreased communication and regulation. So again, improved ATP, improved NAD+, improved levels of peptides like MOTS-c, which is a mitochondrial peptide, which we'll talk about in the peptide class. There's anterograde signals between the mitochondria and nucleus, and retrograde signals. Signals go back and forth. One of the things that I really, really want to make sure that people understand is these things are relatively controllable. Like, they're pretty significantly controllable, you guys. So we can look and study and see that by inducing optimal levels of nutrients and supplements and vitamins and peptides, we can actively change the way that our epigenome is being expressed and that the way our DNA is being expressed. This is one of the proteins that I talk about in the peptide therapy class. MOTS-c protein. It's called short open reading frame of the 12th S rRNA of mitochondrial DNA. It's a long one. It's just called MOTS-c. So this one helps activate the AMPK pathway, which we're going to talk about in a second. It helps activate sirtuins. It helps activate anti-inflammation. It improves NAD+ levels. It improves sirtuin gene activity and sirtuin protein concentration, which helps improve cellular health. All of these mechanisms, to some degree or other, are significantly controllable by what we're putting in our body and how we're stressing our body. And if we understand that, we can do things to combat the detrimental effects of what we're doing. If you understand what your goal is, if your goal is to be healthy, and you understand how you can be unhealthy, if you can just stop those things from happening, which we can do a lot of the time, you can really, really improve your health.